So that's what I've just told you. They arrest the catalytic the two different inhibitors arrest the catalytic cycle at different points. We know what residues are responsible for binding, and we're engineering the protein to try and arrest the catalytic cycle at different points. Now, what about the regulation of the bacterial ATPases? So it's been proposed that this epsilon protein, which is an intrinsic component of the rotor, I'll show you in a moment, the central stalk, acts as an inhibitor. But what there is actually is no explanation of what the physiological role of this might be. It has been shown more or less convincingly now that this protein can assume what are called up and down conformations. And it's been proposed that those two conformations are regulated by the binding of an ATP molecule to this epsilon protein. So there's nothing to do directly with the catalytic sites. It's another site where ATP bound. So we've solved recently the structure of a bacterial ATPase from a bacterium called Bacillus TA2. This is a remarkable organism which grows at pH 10, but it still uses a proton gradient at pH 10 to generate its ATP molecules. So here is the epsilon subunit associated with the rotor. So this is a magenta protein. And here you can see this ATP and actually magnesium bound in this groove in this alpha helical part. So it's been proposed that when the ATP is released, these helices go up and they enter the catalytic part here and inhibit the enzyme. And there is cross-linking evidence that happens. So what we decided to do was to mutate residues that are involved in binding the ATP molecule. So that's the wild type showing how ATP is bound, and these are important residues bound in this groove. So you can see the protein has two domains. There are two alpha helices antiparallel to each other, providing this ATP binding site, and then the rest of it is a series of beta structures. So we mutated those two residues. ATP would no longer bind, and we solved the structure again, and it's shown here. And what we found is that it's still in the down position. So I'm very skeptical, therefore, on the basis of these experiments, that the up and down position, has any relevance to regulation, or that it is, regulated by the binding and release of an ATP molecule. Interestingly, if we superimpose the equivalent protein from the mitochondrial enzyme, which is shown in magenta, in the mitochondrial enzyme, bound in the groove where the ATP would be bound in the bacterial enzyme, is another protein shown in green. It's a very simple protein. It's only 50 amino acids long, and it occupies that site, preventing ATP from binding, if indeed it could do so. We don't know that. But the site it is occupied by another protein in the more complex mitochondrial enzyme. So I want now to come to a discussion of the C rings in the rotor. And so the important point that I made earlier was that the C ring sizes differ between species. Now, first of all, I'd like just to explain how we think rotation could be generated. So here is the C ring depicted in blue in association with the A protein that we don't know the structure of, shown in green. And in this model, it's shown in intimate association with the external surface of the C ring. Now, each C protein, I haven't shown you the structure yet, is a very simple protein. It's made of two transmembrane alpha helices, obviously running in different directions, with their N and C termini on what would be the outside of the inner mitochondrial membrane. So the central stalk in the enzyme is attached here. And in the C-terminal helix of the C protein, which is on the outside of the ring, there's a carboxylate residue right here in the middle of the phospholipid bilayer. So that's a very unusual position to put a charged residue. And according to this model, when the carboxylate in this carboxyl residue in this position in association with the C ring is in association with the C ring, it carries a negative charge, and it can be accessed by a half channel that allows a proton to go in and neutralize it. And once neutralized, the carboxyl, the neutralized carboxyl moves to this position here, which is more hydrophobic, where it's more happy. This then brings another negative charge here. Another protein is consumed, and so you can see that the protons get carried on these carboxyl groups around the ring until they reach this point here in another region of association between the C ring and the A protein, where it's imagined that this site has the properties of making the carboxyl reionize, regenerating the negative charge and releasing the proton on the other side of the membrane. So the path of the proton, according to this model, is it goes in here, it's carried round to here, and then it's released on the other side of the membrane. Now, each time the ring has rotated through 360 degrees, and remember, it's attached to the rotor, you've turned the rotor through 360 degrees, and from the 3 catalytic sites up here, you produce 3 ATP molecules. And so if you know the number of carboxyl groups in this ring and the number of protons required to generate the 360-degree rotation, you know the number of protons required to make an ATP. So, for example, if there were 12 C proteins in the ring, you would require 4 protons to make each ATP molecule. Now, what we found to our surprise was that, first of all, this is in 1999, that the yeast C ring had 10-fold symmetry. So that told us that the cost of making ATP in Saccharomyces cerevisiae was 3.3 protons per ATP. But since then, all symmetries from 10 to 15 have been found in bacteria and chloroplasts. Chloroplasts, spinach chloroplasts, have a 14-fold symmetry, so the cost of making ATP there is 14 divided by 3. In this species with a 15-fold ring, it's actually a cyanobacterium, they pay 5 protons to make an ATP. And so, to many people's surprise, the cost of making an ATP varies between species. So some species are able to pay less energy to make an ATP molecule, and others have to pay more. It was anticipated before this was known that this ATP proton ratio would be a universal number. And it has caused a lot of surprise. But of course, what we wanted to know was what was the symmetry in man and invertebrates. And last year, we found that the ring symmetry in man was 8. And so this told us that the mammalian ring, the mammalian enzyme, was the most efficient ATPase at making an ATP. It only pays 8/3 protons to make an ATP molecule. This shows the structure that we determined of the F1 with the 8-fold ring attached to it. This is the electron density, and there are 8 blobs here in the outer ring representing the C-terminal helix of the C protein, and 8 in the inner ring representing the N-terminal helix. Now, this links to experiments done a very long time ago by Britton, Chance, and Williams in this classic paper published in the Journal of Biological Chemistry in 1955. And this was the first time experimentally that what are known as P2O ratios had been measured with an oxygen electrode. These ratios, and I'll explain what it is in a moment. This ratio had been measured in the 1940s by Ochoa using other methods, but what Chance and Williams did was to make really the first accurate measurements of P2O ratios in mitochondria. Now, if when you feed electrons into the electron transport chain, if NADH is the donor, what they found was that the P2O ratio was in the range of 2.3 to 3, Whereas if they use succinate, then the P to O ratio was 1.5. So here is the explanation of the P to O ratio. It's simply the number of moles of ADP phosphorylated for each 2 electrons that are transferred from these substrates down the electron transport chain to oxygen in the way that I explained at the beginning. Now, there's a very simple relationship between these experimentally measured P to O ratios and the proton ATP ratios that I've described. We can deduce, we think, from the structure of the enzyme. So if you provide electrons via NADH and they're transferred to oxygen, for each 2 electrons transferred to oxygen, that would lead to 10 protons, according to what we know today, being pumped out of mitochondria. When you supply succinate, 6 protons are pumped out. In the case of the mitochondrion, you have to pay another proton to exchange the internal ATP for ADP. And so the proton ATP ratio for the ATP synthase, for the enzyme, is 8/3, according to the structural model, 2.7. But for ATP synthesis, it's 1 more. You've got to add that. And so now you can calculate from these numbers the P to O ratio. It's simply the number, this number here, the number of protons that are pumped out when using these substrates, so 10 or 6, divided by these numbers that you've deduced from the structure. So these are the calculated values, and these are the experimentally determined values determined by Chance and Williams and by other people. And the important point is that the values now are quite similar to each other. And this now leads people to believe that we really do understand the basis of respiration. Whereas before, when we thought that the rings were made through all species of 10 proteins, the numbers didn't coincide so well, and people had begun to think that there were mistakes that had been made in determining proton ratios. Now, this shows you the differences between these various rings. So this is the C8 ring from the bovine or human structure, C10 from yeast. C9 has still not been found, and then there's C11 from a bacterium and C13 from another one, C14 from spinach, C15 from Synechococcus. And then this ring here is a ring where we determine the structure from a V-type ATPase. It's a very high-resolution structure, and it's actually from a sodium-driven enzyme, and we can deduce where the sodium ions bind to the ring in association with the carboxylates. So you can see there's a huge variation in the physical sizes of these rings from one ATPase to another.